Yeah. Just 1 minute, I will start the session.
So we are ready now.
Yeah.
Yes.
Yeah. So, good morning to all of you.
Good morning, sir.
New. Good
Good
morning.
morning.
Good morning. So this will be the, I think, a wonderful session, which is interact with our resource person. In the same way, last webinar, which is on the mushroom cultivation, and today we will go with the another webinar workshop, and it is very related with the research methodology. As per the NEP guideline, it is an essential part for the small research projects, or it may be the any kind dissertation work, so that every student must know how to select the certain kinds of the topic and how the statistics should be incorporated with the life sciences. And for this, today's resource person, Dr. Nilesh M. Mahajan, Professor and Head Department of Pharmaceutics, Dada Saheb Balpande College, Pharmacy, Nagpur. I welcome to you, sir, on this online platform.
Pleasure is all mine.
Yeah. Now I request Dr. Ashok Davande, sir, IQAC Coordinator and Head Department of Microbiology, Associate Professor, Shankar Rao Bhoyer Patil Mahavidyalaya, along with under the aegis of IQAC, Department of Microbiology, Yashwantrao Chauhan College, Lakhandur. I also welcome Dr. Rakesh Thakre, Head Department of Microbiology, Yashwantrao Chauhan College, Lakhandur. And on the behalf of IQAC and both the colleges, Shankar Rao Bhoyer Patil Mahavidyalaya Pulgaon and Yashwantrao Chauhan College, Lakhandur, I welcome today's resource person, Dr. Nilesh Mahajan. Now I requested Dr. Ashok Davande, sir, please give the welcome remark.
Thank you, sir. Am I audible, sir?
Yeah.
Good. Good morning, everyone. Welcome all. It's my great pleasure to welcome our esteemed resource person, Dr. Nilesh Mahajan, sir, Professor from Dada Saheb Balpande College of Pharmacy, Nagpur. Today, we gather here on this workshop, Research Methodology. This workshop is on research methodology is basically the systematic way of finding the answer to the questions or solving the problems. This workshop helps us to plan our study, collect data, analyze it, and draw meaningful conclusion in a systematic manner. Research is not just about gathering information, but about doing it in a right way. And that's exactly what research methodology teaches us. We are truly fortunate to have Dr. Mahajan sir with us today. Dr. Mahajan sir, highly respected academician with rich experience in teaching and research. Today he is here with to guide us on an important topic, that is research methodology, and I am confident that his knowledge and experience will greatly benefit all of us. On the behalf of institution, IQAC, and Department of Microbiology, I warmly welcome you, sir. Thank you for sparing your valuable time with us. With this, thank you very much. Sir, over to you, sir.
Thank you, sir, for your remark. Before start the session, it is my duty, it is my pleasure to introduce today's resource person. I worked near about 12 years with sir at Akola Institute of Pharmacy. Dr. Nilesh Mahajan, sir, is a distinguished academician, researcher, and innovator with over 24 years of teaching and 19 years of research experience in pharmaceutical sciences. He is currently serving as a professor and head department of pharmaceutics at Dada Saheb Balpande College of Pharmacy, Nagpur. Sir, research expertise and guidance, expertise span in nanotherapeutics, crystal engineering, inhalation formulation, and polyherbal drug delivery system. Sir guided 83 postgraduate student and 4 PhD scholar to completion, contributing to the capacity building in the field. Sir has successfully evacuated external funding research project worth rupees 1.2 crores from the agencies including AICT, DBT, and BARCS, and extended industry consultancy generating nearly 1 crore in funding. Mahajan sir has an impressive patent portfolio of 13 innovation out of 3 international and 10 Indians, covering novel drug delivery devices, nanostructure formulation, and medical devices. Some of his notable inventions include robotic jaw for in vitro dissolution testing of chewing gum, nose-only exposure chamber for preclinical assessment of dry powder inhalation, pH sensing, swallowable devices for the gastroesophageal reflex disorder, automated spreadability tester, and film former. Sir publishes around 75 peer-reviewed publication, contributing the several book chapters and edited as in the elsewhere book Phytophysics and Nanophysics in Therapeutics. His scholarly work has established a strong interdisciplinary collaboration with organizations such as DRDO, Nagpur Veterinary College, Lakshmi Narayan Institute of Technology, Nagpur, Govigyan Anusandhan Kendra, Poultry Research Center, Government Dental College, Ramdev Baba College of Engineering, Raisoni College of Engineering, etc. Dr. Mahajan sir is also recognized as a resource person in more than 30 conferences, workshop, and faculty development program, delivering an invited talk on targeting HIV reservoir by dendritic carriers, co-processor exhibitants, ethics in publication, sociologist, innovations, and translational research. He has organized several national and international scientific events in collaboration with CRSIC, IPTI, IPA, and IPC. Sir is a life member of APTI and executive member of Control Release Society Indian chapter. He has presented his research globally, including nominated a speaker at Control Release Society annual meet Italy 2024. His contribution has been recognized with the numerous award including the Best Researcher Award 2022 by Rashtrasant Tukdoji Maharaj, Nagpur University, Nagpur, along with Dr. P.D. Patil National Award of Best PhD Thesis 2024. Through his pioneering research, strongly industry academia connect and committed to the innovation, Dr. Mahajan sir contribute to inspire the next generation of pharmaceutical scientists with advancing the frontiers of drug delivery, research, and translational health care. This is a very brief and a short biodata of today's resource person, Dr. Nilesh Mahajan sir. Over to Mahajan sir for rest of the session on research methodology.
Thank you, sir. Thank you so much. I hope I am audible to all of you.
Yes, sir.
Yeah,
Yes.
sincere regard to Dr. Yogesh sir, to whom I worked as he mentioned almost for 12 years, 12 long years. We have fond of memories at Institute of Pharmacy, Akola, and I learned a lot from him. He being a core academician and a very learned person. I owe sincere regards to Dr. Ashok and Dr. Rakesh, who are there meticulously working for the successful organization of this workshop, along with the Dr. Yogesh sir, being a part of IQAC activity, and I can see the wonderful integration of academicians over here now, and PhD aspirants are also, as he mentioned earlier when we were discussing this gathering and this particular dissemination or the program workshop for this dissemination of the knowledge. Of course, this is fantastic to see the PhD fellows along with the PG fellows, and the some UG fellows are also there. As he mentioned, with the advent of the new curriculum, and UG students also have to attend, or they shall undergo the project. So this will be for this particular 40-45 minutes or 1 hour will be a wonderful journey for all of us, and we could actually learn from each other. Of course, this is a very basic topic I'm covering over here. As I was not aware that UG fellows are also there, and PG students as well as the PhD students, because on the several forum I do have a different topics to discuss. Like he mentioned that how to select the outcome oriented research from that point of view, up to the publications, and altogether up to the presentations and the some proposal writings also. So sooner or later, maybe in the next week, or maybe after in the next month, I would like to deliver on those topics. That's a different part. So today's agenda, of course, is a basic research methodology. I hope my slides are visible, sir.
Yes, sir. Sir, 2 minutes. I will stop you. Firstly, I will congratulate you. You are clearing the MPSC examination with the highest percentage in the interview. So
Thank
for
you,
that, really, you are appreciable, and congrats.
Thank you, sir. Thank you so much. I request admin to please, this annotation is going on the slides also. Some people are annotating on that. So please stop
Okay.
that. And again, I request all of you to please be muted. And of course, it would be better to have camera off for the rest of the guys. The organizers will be there along with me. Of course, if you have any question, you can put in a chat. So I can see one annotation on slide itself now. Can you please do the needful, sir, to avoid
Yeah.
such nuisance? There is one line drawn by somebody. I'm not
Yes, sir.
getting it.
But yes, sir, please remove that. Your host now.
Yeah, yes. Just a minute.
Yeah. So admin can do it to just stop the annotation.
Yeah.
Here is it, sir.
It's okay? Yeah.
Sir, thank you.
Yeah.
Fine. So let me straight away start with the discussion on the research methodology. See, research, as far as research is concerned, it's a vast terminology, and it could be difficult to cover it in a any point of time, or any conference or workshop. If you are holding a workshop or conference on the research methodology, of course, there are so many modalities around the research methodology that more often than not we discuss on the different forum. Right from the beginning, as I mentioned, the selection of the topic, then the cordial relationship or harmony between the fellow and the supervisor, and of course the planning, the execution, the result, the data generation, and the publications, or the generating some IPR around that. So research methodology is a vast topic, I would say, and of course it's an umbrella term. Research cannot be defined in terms of qualitative or quantitative means. But let me try to sum up with some of the important modalities as far as the basic research methodology is concerned. So yeah, transition is happening at your end, sir. Slide transition is there. I now move to next slide. Is it visible, sir?
Yes, sir. Yes, sir. Go ahead.
Yeah. So transition is happening over there. Fine. So let me take you all to the journey of research methodology, the basic components of the research. Of course, how could I go straight away to the modalities before introducing you with the research? As I mentioned, it's a really difficult to sum up the research within a 1 or 2 slides. But let me try. It's a systematic and scientific process, as you all might be aware about. And of course, we will every researcher aim to discover the new facts, or verifying the existing knowledge around any domain for that matter. So it will help. Research will also help in solving the practical as well as the theoretical problem day in and day out, and it is widely used in the every field, not restricted to the science, basic science, or the pharmacy or the medicine, but covering all the domains, etc. As you better know about that. So what I mean to say, when we talk about the research, the first thing that might be coming into the mind, that this everything, I mean, whatever there with the innovations and the research, more often than not we talk about the startups nowadays. But as far as the startups are concerned, startups are driven with the innovation, and innovation is driven by the research. So basic thing is the research. Once you have a research and you have some technology ready products, or technology ready for the translation, then and only then you can think about the startups and the entrepreneurship, or other modalities for that matter. So what I mean to say, if you think about the research, quickly the things starts revolving around some research products. Basically, maybe you better know about the COVID-19 vaccine. As I'm a pharmacy background academician and researcher, I'll be more giving more examples of drug delivery and the some products which are available in the market, pharmacy based product, of course. So as I mentioned, COVID-19 you better know, and wherein the scientists managed to develop a vaccine in the record break time. Okay, and if I'm not wrong, it was not a luck. Okay, of course, even though they took very short period, and it was developed in a very record break time, the scientists strived, and they do their best and they went with the structured research methodology. It was not possible without following the structured methodology. If you are not following that structured methodology, you won't be able to get those results, those kind of result. And believe me, if you refer to the other vaccines that were developed earlier, it will take around 5, 6, 7, 8 years to develop a particular vaccine. But in the record break time, India managed, the scientists and the of course formulation development scientists, they managed to brought that vaccine with the record break time. It's because of their structured way, because they understand that problem meticulously, and they proceed with the systematic and structured manner to follow. And of course, that's why I'm insisting you and giving this example. Even though there is a strong team across the different having a different expertise, but still you people should work with having some common goal, or the some research goal over there, and it has to be followed with a structured methodology. So of course, why I'm telling you, because research cannot be defined within a context of any particular domain, but it can be applied to pharmacy, medicine, science, even for that matter. It's a systematic and scientific process. So systematic process, it is of course it is a systematic process, and we may develop some product, we may discover some new facts, or there might be some existing gap in the knowledge that can be verified by doing some practicals, and probing data can be data can be generated by doing the probing experiments, and of course theoretical as well as the practical problem can be resolved for that matter. So definition of research, let me tell you that these are some guys, the renowned academicians Redmond and Mori and Clifford Woody. If you read books, core books of the research methodology, you can see this definition over there. It is a systematized effort to gain the knowledge. Of course, this research has been developed, defined by the Redmond and Mori by in the terms of efforts, and to gain the new knowledge. Of course, you will not get the knowledge without the systematic efforts to create some data, or to generate some data, to get the knowledge and to disseminate that knowledge, of course, by doing some publications for that matter. And again, Clifford Woody, I read this again in the research methodology book. Careful inquiry or examination of any particular issue, I would say. So careful inquiry, as I mentioned, the experiments has to be planned and executed to generate the data, and of course we will be evaluating all those data for that matter, or all those development, whatever you have developed. If you refer this definition, of course, one thing is very clear. The bottom line or the takeaway message must be, research is a search for the knowledge. If you ask me any example, like for a scientist who is working on some multi-drug resistance antibiotic having a multi-drug resistance, and you're finding the way or to tackle the MDR, multi-drug resistance. Why I'm giving this example, because this is very relevant in the context of the pure science. Or else, if you would like to develop a mRNA vaccine, okay, it's hardly possible without experimenting, and if you are randomly experimenting, you will not get the results, of course. So systematic effort has to be there, and systematic effort to gain that knowledge, to mitigate that gap which is in existence, because as I mentioned, several bacteria are having a resistance across several antibiotics. So by one or more experimentation, or doing some conjugations with the polymers, maybe some nano carriers, you can do the research around that, and you could come out with some strategy that could be able to mitigate those gaps. So this is how research will be driven in a search of a knowledge, okay, as far as the definition is concerned. Then as if you ask me, because we are here to discuss the research methodology, and of course, as I mentioned, that it is hardly possible to discuss the research methodology without actually knowing the research, what is research, and what are the other modalities around that. So research versus research methodology, if you ask me to compare, research I believe is actual investigation process for any product, development of the product, development of the technology, or mitigating any gap over there which is in the existing market. So this is all about the actual investigation process as far as the research is concerned. However, research methodology is around the study of the methods that could be used to conduct that research, actual investigations that I am talking about. Okay, so it includes the tools, different tools of the research, of course, the different techniques that can be used, and different procedures to generate the data. So this is why we are following the research methodology because there is a systematic planning, there is a way to execute, there is a plan of work, there are some tools that can be used. Of course, there are some so many techniques and the procedures that are available in the literature, and by doing those probing experiments, you will get the data, and then you are coming to the conclusion that this is my investigation around that particular case. Okay, so as far as research methodology is concerned, the flow is quite simple. Defining the generating the hypothesis or defining the research problem, of course, is the first and foremost task. Then going down, developing an approach to define that problem. Of course, what is the origin of the problem, from where it is coming, what is the prevalence. If you are talking about the, as I have given example of a multi-drug resistance, how prevalent it is around the different corners of the world. And as far as India economy is concerned, socio-economic burden, healthcare burden, all these things needs to be taken into the consideration while generating the problem. Formulating the research design, then the next part is formulating the research design, which is very, very important, because if you are following the formulating the research design meticulously, then only it is possible for you to collect the collect and validate the data. So it's not only about the collection of the data, but it's about the validation of the data, because whatever data you have generated and you are sooner or later you're going to publish that, somebody will follow you, and if he or she will not get those kind of results, of course there will be a gap, and then they said they will be coming to the conclusion that whatever data has been provided by a particular XYZ scientist is not correct, is not validated, because it is not reproducible at my end. So this may be the possibility. So whatever design you are formulating, it should be robust enough that everybody can get the reproducible result with that data that you have collected. So fourth process, collection of data, validation of data, of course, preparation and analysis of the data, as I mentioned, and finally reports preparation and the presentation. This is how research methodology will go step by step. So methodology is simple: how to do the research across that particular case, or to achieve the investigate some facts or some gaps, or to study that domain. So research is about actual investigation process, and methodology, of course, is about how to do the research for those investigations, as I mentioned earlier. So if you ask me that there are so many terms that we often use, more often than not we use about the discovery, the invention, the innovation, but these are some confusing and misleading terms in terms of their exact origin, in terms of their exact meaning. For example, if you are talking about the discovery, finding some new things, of course penicillin was discovered way back. And if you talk about the invention, creating a new product, you're creating a new process, creating a new technology, can be called as an invention. And as far as innovation is concerned, of course, you better know about the improving the existing idea. Whatever ideas that has been already in the public domain, or whatever gaps are there around that product, around that technology, around that process, you can always there is a chance, there is a scope, there is an opportunity to deal with that, and you could come up with some improved idea, much better idea, much refined product for that matter, and those are referred as innovation. So there is a clear-cut distinguishing features when it comes to the discovery, invention, and innovation. Discovery, of course, finding something which is in existence. Okay, finding something new and publicly make it announced but of course it was there in the existence. Okay, that is the discovery. In invention, as far as invention is concerned, creating some new products or the process or the technology, as I mentioned. And innovation, of course, is improving the existing ideas or improving the existing products and some technologies. Okay, so all are outcome of the research. If you ask me, sir, why you are discussing, because these are the outcomes of your research. As I mentioned, even if you would like to develop your product and launch it in a market, and you would like to start your career with some startup, with entrepreneurship, you need to do the research. Okay, so that research will drive the innovation, and then it will drive the startups, as I mentioned. So this is very common nowadays to do some research around some development, and creating that or creating that intellectual property, protecting that intellectual property, and going for the technology transfer. That is what is expected from we as a researcher, we as a scientist, and you people as a young scientist working on the different domains, because our country, as we know that it's a developing country, and we would like to match up with some of the big countries like China, USA, or some innovation driven countries. For that matter, we all should come together and have a discussion around, and we need to set up a forum, and we need to have a collaboration for those kind of technologies or the product that can be licensed, and more and more products can be coming out into the market. So that should be our dream, of course. Then let's move on to the characteristics of the research. As you if you ask me, there are some predominant characteristics, like it should be systematic and organized manner. It has to be done in the systematic and organized manner. It should be very logical. The objectives must be clearly defined. And empirical, it should be empirical. That means it has to be based on the observation. Whatever you are claiming, whatever data you have, it should be based on the observation. As I mentioned, it should be reproducible, okay, to any lab, anywhere in the world, and it should be controlled and critical for that matter. So when we talk about the characteristics, of course, if you refer any example, any anti-cancer formulation is tested, results must be reproducible across the lab globally. If you are preparing some nanoparticles, and it is it is working or giving the results in only in your lab only, but fails to reproduce elsewhere, then of course it's not a reliable research. Okay, so when we talk about the characteristics of the research, the research must be systematic. This must be organized, and logical steps has to be followed. Objectives should be very precisely defined, and of course it is based on the observation, result collection of the data, validation of the data, and reproducibility has to be there. Okay, so if you follow these characteristics meticulously, judiciously, I would say, then there is every possible chance that sooner or later you are going to create a best product, best technology, the best science publications or the patents for that matter. So you have to follow these characteristics, which are very, very important. Then I'm moving to the types of the research. You better know about that. I will not go into the very details about that. But in healthcare system, if you ask me, because as I mentioned, I being a guy of or integral part of the healthcare system, being a pharmacist, we rarely rely on just one type of the research. However, sometimes it's a combination of the different types of the research. Even developing a drug may involve a fundamental research. Okay, it's a part of a type of research, one of the part of the research. Thinking about the chemistry, complex chemistry, generating the new molecule or preparing the new molecule, drug discovery. Okay, and of course, it's a fundamental part of a fundamental research, and its impact onto the some kind of a disease, some indications, some elements, or the disease pathway, I would say. So this is about the fundamental research. And applied research, of course, creating some formulation out of that drug, which has just developed by some lab. So formulation development, I would say, it's a fundamental, sorry, it's applied research. However, molecule synthesis is maybe a fundamental research, is a part of fundamental research, or understanding the disease pathway may be a fundamental research. Of course, that develop a molecule being under the fundamental research as a type of a fundamental research, then will develop a formulation for that drug. It's coming under the applied research, and finally we are testing it clinically. Okay, so you are doing a clinical research on, and which is again a part of a qualitative and quantitative research. So single topic have so many types of research. It's not like that I'm doing only applied research, I'm doing only analytical research, I'm doing only a descriptive research. It's not like that. I strongly believe that one single topic could have so many types of research, and researcher has to attain so many types more often than not. Okay, so if you first ask me to compare a descriptive versus analytical research, it's very simple. Like descriptive is about the, it will describe the current situation, whatever prevalence is having. Like for example, if I would like to work on some nano targeted delivery for triple negative breast cancer. Okay, so it's during the early days, I need some data. Okay, as for the prevalence of that triple negative breast cancer, how many number of women they are affecting with the TNBC, for that matter, whether it is coming out of the gene or some other other matter is about the concern. So other thing are involved in that or not. What I mean to say, it's a describing type of research, which is we call as a descriptive research. For example, prevalence of the disease, as I mentioned. Then analytical research will existing data needs to be analyzed. For example, any effectiveness of a drug, or any drug effectiveness study, for that matter. So if you ask me, I can give example of diabetes or any disease like as I mentioned, triple negative breast cancer, for that matter. So we can analyze any drug which is available analytically. We can have a PK, like pharmacokinetic, pharmacodynamic data, and of course effectiveness study around that. So this analyzing the existing data will be the part of analytical research, of course. If you ask me about the applied and fundamental research, as I mentioned earlier, that practical problem solving, like for example, drug formulation may be a part of applied research. Fundamental research, as I mentioned, maybe a mechanistic study, maybe some theoretical knowledge generation, or some if you ask me about the any example with the pharmacy background, maybe discovering some molecule across that. So fundamental research this is the part of a fundamental research. Quantitative versus qualitative, of course, you better know about that. You might have know about the non-numerical data, numerical data, statistical analysis, as Dr. Yogesh sir was telling you in the introductory note, that we should understand where to apply the statistics, what kind of statistic has to be applied, where to apply the one-way ANOVA, two-way ANOVA, t-test, and all these tools, statistical tools needs to be applied, and all these are the part of the quantitative data generation, and it will be a part of a quantitative research. However, quantitative research, of course, is relied on non-numerical data. There will be no numerical data, for that matter. Maybe getting some data out of the interviews, some podcasts, some observations, some statistical survey. So all these will be coming under the qualitative data, not the quantitative data. Of course, if you have some experimentation done with for any case, for any technology, for any product, or any scientific term. So for that matter, if you have data, then the data has to be tested statistically, and quantitative data has to be generated. It is the quantitative research, and I believe we as pharmaceutical sciences, basic sciences, engineering sciences, and medical sciences, all are doing the quantitative research more often than not. Again, conceptual versus empirical research. Conceptual research will be based on the ideas and the theories more often than not. You will have a like for example, CRISPR-Cas technique you might be knowing about. It's about the having a tested the CRISPR gene editing technique tested in the lab, and it existed as a concept. Of course, it's a concept, and that has to be tested. That conceptual research can be done, and one scientist start his study editing the gene. Okay, and in the cells, or observing the results, it becomes empirical research, of course. And these are the two sides of the coin. Like if you ask me, conceptual research and the empirical research, the concept has to be there, and that has to be tested. Okay, by doing some experiments. So the concept we know, how bacteria are developing resistance. Okay, that concept we know. But to be very frank, to generate the data, you have to do the empirical research, because it will be based on the experiments and the observations. So you will start working on the basis of that data, the concept that how the bacteria are developing resistance. You know about the several mechanism of bacterial resistance, and now you have started working on one or more mechanism to mitigate those mechanism, to counteract those mechanism. You need some experiments. You have to do some experiments. You have to generate the data. You have to have some observations for that matter, which is a part of empirical research. And the concept from where it is coming, it is a part of a conceptual research. As simple as that. Okay, then steps in the research and research progress or process. Of course, there are so many steps, of course, and each step is having is its own importance, I would say. So conducting a literature review is the first and foremost thing. Why it is important? Because you will get so many you will get a knowledge basically by gathering that existing data. Okay, onto the topic. The topic will be very you will get the more clarity on the topic by reading some articles which are there in the public domain, which are manuscript which are already published in the several journals, reputed journals. So I'll always recommend my student to follow some good journals. Having said that, maybe elsewhere, maybe a Springer, maybe a Taylor Francis, such agencies which are indexed with the Web of Science, wherein you will have some reliable data, because these all are peer review journals, which are the agencies which I am telling about, the elsewhere, the Springer, the Wiley, the some other also Taylor Francis for that matter. So they are actually involved in a very rigorous process of publicizing the manuscript, and they always follow the peer review system. So whatever publications we are referring, it should be having a high quality. Maybe a Q1, Q2, QT, Quartile 1, Quartile 2, Quartile 3 journals are there. Ranking of the journals are there, which is mentioned with the Clarivate Analytics, and you have to follow those. Only then you can generate your hypothesis by reading those publications, and of course you will generate the knowledge around the existing data. Whatever the data it is there in the public domain, you will have a better knowledge and better understanding of your project, and then only you can formulate the hypothesis. So as I mentioned, it's for example, if you are one of my PhD student is working on the epilepsy. Okay, so we are developing one new product for epilepsy, nose to brain targeting. We are just simply developing one spray which can be administered nasally or through nose, and then that drug will directly reach to the brain. Okay, to mitigate that challenge of epilepsy. So there are so many drugs existing in the market. Okay, but all patients will not respond to each and every drug or any delivery system for that matter. So strong research problem can be generated out of this knowledge, because from where I get this knowledge, I get this knowledge by studying the marketed products. I gain this knowledge by referring to the several manuscripts, several patents which are around the corner, and then I have generated the hypothesis that yes, I can improve this delivery system across the blood-brain barrier. I can go through these portal delivery, which is ready-made channel available, nose to brain. I can target the brain through nose only, and I can develop a spray containing the neosomes, that is a non-ionic surfactant nanocarriers, which will be very effective for that epilepsy. So why I am telling this example, particularly you being a science student, there are so many examples you can put forward and discuss around with your mentors. Only thing what I would like to tell you about, unless and otherwise you have a better reading, you have a so many reading of the publications which are there and the patents and understanding the market products, you would not be able to formulate the hypothesis. Let's come to the next step, that is the formulating the hypothesis. So proposing the potential explanation or predictions is what I believe is a formulating the hypothesis. a vague ideas are so many. I mean, when I discuss with my colleagues also, and on a different forum, when I meet to the students, the fraternity, so many ideas are there with so many young aspiring minds. Okay, the researchers, even faculty members, even some research fellows. But of course, all ideas are not viable. What I mean to say. So if you ask me the question, any example, I would tell you about the cancer treatment, for example. So treating a cancer and for generating a hypothesis around that is not uh, the it's not a fully focused. I would say the problem has not identified properly, and the scope is very wide enough. So that vague idea cannot be formulated as a hypothesis. So it will not be enough. But redefining it, and as I mentioned, improving the cancer treatment. If you are formulating a hypothesis like improving the cancer treatment, it's not a proper hypothesis because it lacks clarity, it lacks focus, and the scope is very vast. And of course, you will not get the outcome around that. So you should redefine it by reading the papers, by reading the manuscripts which are available in the public domain, developing a targeted nanoparticle with a ligand attachment for the triple negative breast cancer. That could be a good hypothesis. Why? Because all corners has been ticked, and all the corners has been assigned. I would say it's a having all the boxes tick. Why? Because what you are doing to what you are going to do, I am developing a nanoparticle. For what matter? I am for the triple negative breast cancer. So indication is there. Okay, indication is should be there, or disease condition you have already given, and you have given a nanocarrier system. Then ligand attachment, again clarity on that, how you are going to target. Simply passive targeting is not happening over there, or it's not going to serve that purpose. So active targeting, I am doing it. So all boxes are ticked. So this time kind of a formulation, hypothesis has to be formulated, wherein the clarity must be there, and based on that only you can generate your objectives. Because if you have generated a good hypothesis, then only objectives can be derived. If not, it's very difficult to derive the objectives for these kind of a hypothesis. So hypothesis should not be vague; it should be very clear, precise, and specific. Okay. And it should indicate actually what you are going to do within that research domain. So that is about the formulating the hypothesis. Then designing the research methodology is all about outlining the plan for collecting the data and analyzing the data or validating that data. So there are two attributes which are most important within this particular case, that designing the research methodology. The first one, outlining the plan, that is the plan of study, your study, how you are going to proceed. Okay, that is your plan of work. Should be very meticulously designed and judiciously follow for collecting the data and analyzing that data and validation of that particular data. So designing the research methodology is very important. Believe me, it's a major crux. Okay, as far as your research is concerned, if you miss any of the plan, within the plan, if you are miss any of the any method method for the development or an analysis of that data, you will miss the trick, and then you will eventually be ended up losing all the crux of your research. And then you start following somebody, and then you realize that अरे यार ये तो मेरा रह गया था, how could I forget to do that, and then I don't have a time to cope up with the situation. Then suddenly you stuck up, and then you will find no way to come out of that situation. So whenever you are formulating or designing the research methodology, you should consult to your mentor. Not only mentor, if you are any collaborations or collaborators around you, so you should sit together. All should sit together and have a brainstorming session around what shall be done and what has to be done, and what shall be avoided and what not to be done, and from where I'll get this help, like feasibility, in terms of feasibility also, practicability, all together, right from the material that you are going to use, techniques, the equipments, the resources available or not, everything has to be planned. And then you will get you will have a proper design research methodology for collecting the data. Let's move on to the collection of the data. Of course, gathering the relevant information through the probing experiments that has been conducted. Different experiments we conduct to get that data, and of course that data has to be analyzed with some some or more technique. Okay, so interpreting the findings is very important. If you are getting the data, XRD आपको आपने किया, DSC differential scanning calorimetry, या for that matter SCM आपने किया, so it's very costly. Okay, so your time as well as your money has been already invested. So if you are not interpreting that data, the findings very meticulously, if you can take a help of some expert, or you can take some expert opinion also. Like for example, I'm not a guy of microbiology. If I'm stuck up with some data, I need to go to the I need to approach to the Dr. Yogesh, who can help me for that matter, or else if some pharmacology guy is around there, and I would like to discuss some pharmacological mechanisms and all these things. So XRD, DSC, so altogether you can have a liberty, and you should rather, I would recommend all of you to approach the different researchers having expertise in that domain. By doing this stuff, what you are doing, actually you are nullifying the errors. Okay, because most errors are happening with the first thing, while generating the data, doing the experimentation, and after collecting the data, interpretation. So you have to have some good collaborators around you, and having good expertise with the different domains, and you can approach them. You should not hesitate. You should not hesitate to go there, and don't bother about what he or she will be telling with my data, and whether they will be stealing my data. ऐसा कुछ नहीं होता है, हमारा ये सोच होती है. तो ईगो थोड़ा सा बाजू में रख के हमको अप्रोच करना चाहिए लोगों को, and then you suddenly will realize that my findings are very good, and I have a better understanding. I still remember while doing my PhD, I was not able to read the histopathological slides and understand and interpret those slides. So I straightway went to the one pathologist, Dr. Ingole, from veterinary college. I went to him, and he explained me, Dr. Pathak and Dr. Ingole, I still remember, who were there in the Punjab Rao Deshmukh Krishi Vidyapit, being a part of that veterinary institute. And then they tell me how to read those slides, how to understand the various inflammation processes that are going to that was happened with my samples, with the colon samples of the rat के जो colon के sample थे, जिसको histopathology मैंने किया था. So I was not able to understand that complexity around those histopathological slides. So they explained me, and I got so many clarity about that, and I was very fortunate that I went there and I understood that. And it's very good for your defense also, because eventually you have to present your dissertation before evaluator. So if you are doing that stuff from some other guy, okay, and if by sending that data and just getting that interpretation will not serve the purpose, will not suffice, because you have to sit with him or her, and then you have to understand by your own कि सर ये ऐसा क्यों होता है, ये अगर डेटा इंटरप्रेट कर रहे हो आप, तो ये किस बेसिस पे कर रहे हो, मुझे समझाओ. And once you have understanding, proper understanding, it would be better for you during your defense. You could able to defend that, because so many questions will be asked around that corner. ये आपने कैसे किया, how you followed that, how you get those results, how you analyze those results, how you interpret this, what is the meaning of this peak, what is the meaning of that peak, how you have interpreted this histopathological slide, inflammation कहाँ पे हो रहा है, यहाँ पे inflammation नहीं हो रहा है. तो ये सारी चीजों को समझना जरूरी होता है. Once you have a better understanding with the help of some expertise, then only you will be able to defend it properly. Okay, so let me move forward further. As far as research problem is concerned, we have already discussed it, that it should be relevant enough to address a significant problem. See, generating a research problem, it is not a big deal nowadays, because if you refer philosophy of the government, you will find n number of topics with some prompts from the government itself, because there are so many thrust areas that they prompt through the several websites. Like for example, if you refer ICMR, if you refer any DSC, DBT, whatnot, you'll find some thrust areas over there. So government would like to have a more and more researcher, and they would like to give the funding to to those researchers who are working on that particular domain. So you can have a clear-cut idea, and you will have a liberty, and you will have, of course, you have a good opportunity to work on those different areas of thrust areas, I would say, of a government. So relevant area, or otherwise, you should be vigilant enough to gather that significant problem from in and around, when we talk about the day in and day out routine. For example, waste management, solid waste management is a huge problem, so you can work on that. Okay, routinely you find so many solid waste around your society. So societal innovations you can do, of course, mitigating the challenge of MDR, multi-drug resistance. Okay, bacterial biofilm, for that matter. So there are so many relevant research topics can be generated, of course, which are having a societal impact, socio-economic impact onto the our with our society, our country. Thrust areas prompted by the government. So all these are the significant problem, relevant problem, I would say, and you can take any of those problems, of course, for as a research problem. But again, the second thing which is important is it should be measurable. Whatever data you are generating, of course, it's a part of a quantitative research. So you will have a quantitative. You have to allow yourself for the quantitative analysis, and the research problem should be specific to ensure the focus particular issue only, not very vague. As I mentioned while generating the hypothesis or creating a research problem, so you should be very specific based on the any particular disease, any particular phenomena, any particular case study, for that matter. But it should be specific enough for the getting the good data and disseminating that knowledge. It should not be too vague, because there will be a time limit again for MSc guys. I don't know how many months you will get, or you are getting, for that matter. PhD guys, more 24 months or more than, not more than that. So of course, looking at the timeline, you should be very specific, and you should not be too vague, because if you are too vague, eventually you will end up with the so many data, but having any without any significant impact. Okay, so don't do that. So formulating a research problem, of course, is just like a planting a tree. Okay, because there will be a nut, and then you are sowing that seeds, and the seedlings will be coming out, and so slowly and steadily it will be developed. So why I'm saying that, why I'm giving this example, because once the idea is in your mind, you need to discuss it with your mentor, and mentor, of course, having his own setup, having his own resources, having his own ecosystem. So with the help of that ecosystem, with the collaborators, and there should be a research family, what I strongly believe. It will start from the mentor, then PhD students, then PG students, and the undergraduate student. So you should build that family with you, and then you have to sit together, identify the research area, narrow down the scope. Okay, narrow down the scope is very, very important. As I mentioned, it should not be too vague, and define the objectives. Once have a clear cut hypothesis you have generated, then it would be easy for you people to define the objectives. I'll tell you one example. Just now I have submitted one ICMR project, as I mentioned earlier, wherein they are actually asking for 2, 3, or not more than 4 objectives. So whatever you are proposing, I propose the gene therapy over there, and only 4 objectives they have asked, not more than that. So why they are asking only 4 objectives? Because it will be sufficient enough to suffice to justify whatever the topic you are proposing. So objectives should be very particularly defined, and those objectives should be very not in a huge number. 2, 3, 4 at the max, it's sufficient enough. Okay, I somewhere read in some dissertation that procuring the material is one of the objective. it should not, it could not be a, how it could be a objective. I mean, objective is always revolve around your the research methodology, okay, or or your formulation problem, or your research problem, I would say, to be more precise. Okay, so this has to be done very peculiarly, very particularly, and you have a particular objectives. As I mentioned earlier, you should have some feasibility checks also. कि हमारे lab में ये equipment है या नहीं है, whether we have that equipment, whether we have that chemical, whether we have that biochemical kit with us, what is the costing of that biochemical kit, whether do we have an animal facility, if not, to whom I can collaborate. Okay, so feasibility check is very, very important. Again, I have a HPLC with me, whether the solvents will be affordable to me, because this is a self-funded project. It's I don't have any funding for that matter. So feasibility check is very, very important. While formulation of the problem, you should always ensure the feasibility at your lab. If not, I'll tell you the best possible option that you can approach to other collaborators, the other labs, and of course, don't curtail your for objectives or your research project because of the unavailability. Okay, those days are gone. आपके पास सब कुछ होगा, जरूरी नहीं है, इसलिए जरूरी नहीं क्योंकि सारे लैब्स फुल्ली इक्विप्ड नहीं है, या मेरे लैब में जो मुझे चाहिए वही सारे इक्विपमेंट रहेंगे, आपके लैब में जो आपके रूटीन के लिए, करिकुलम के लिए चाहिए वही सारे इक्विपमेंट रहेंगे. सो इट वुड बी हार्डली पॉसिबल टू हैव ऑल द फैसिलिटीज इन अ सिंगल लैब. सो इट वुड बी बेटर to collaborate around for the different type of study, for the to generate different data around in and around your research project. So don't skip that. Because curtailing your research project because of the unavailability will narrow down your topic, and of course it would be so insignificant that eventually you will end up with a very less data that cannot be published in a reputed journal. Okay, so this is about the formulation of the problem. Then of course I'm coming to the literature survey. As I mentioned earlier, that it's a first important thing, a very first thing that all researchers are actually they do while performing any research project or doing any research project. So as far as this literature survey is concerned, you can see reviewing the existing study is very, very important, and from where that problem is coming out, that background knowledge should be sound enough. You have to read some books for the basic data or basic procedures involved. Then refer the several websites, understand the dynamics, whatever current happening is there with respect to that topic. If you don't have that idea, then again it would be difficult to generate a proper hypothesis or planning your study. And unknowingly, there is a possibility that you have formulated a problem which is already there in the market. So it's none of your use. So market dynamic has to be understood, and through the website you should study different websites or Google. You can simply Google it out, and you get the knowledge what is happening in the world across that problem. Okay, avoid duplication. If you are doing the duplicate study, of course it's quite insignificant in the context of a publication, and of course you will ended up with publicizing that in any substandard journal. So that can happen if you are doing a duplicate study, or duplicate generating a duplicate data which is already available in the public domain. As I mentioned earlier, you have to use good quality journal. Having said that, always refer WOS index journal, or the journal having the Q1, Q2, Q3 rankings, quartile 1, quartile 2, quartile 3 rankings, having a good impact factor. Of course, there are so many agencies, as I mentioned, Elsewhere, Springer, Taylor Francis, and all these things. Okay, so you have to use those journal only, because as I mentioned earlier, these are peer review journal, and whatever data they have published can be reproducible in your lab. And of course, as I mentioned, if you are doing some innovation around, and your research is innovative, you can generate the patents out of that. So a literature survey literature survey has to has to do across all these things like existing study review, generating the background knowledge, studying the books for basic things, websites, just to avoid the duplications, referring the quality journals, and referring the patents also. If you are doing some innovation, you have to refer that those patents which are already there in a public domain. So Google patent you can search always, or US patents by referring to the uspto.gov.in, or Indian patents can also be referred for that matter. So literature survey, before starting any research, scientists or researcher should spend months together reviewing the existing work, as I mentioned earlier. Okay. Then importance of the literature survey already we have discussed. So I'll skip this slide. Then primary and secondary sources. When developing a new drug delivery system, for example, I'll give you original research papers are primary sources. Okay, for that matter. A review article, if you are doing a review article, summarizing the multiple studies which are always there in the public domain, which we can refer as a secondary sources. Okay, so both are essential. Always, I recommend my students to first read the books for getting the basic idea clear. Then I always insisted them to go through the review articles, because review articles will give you the systematic approach and the data which is always there in the public domain, and the researchers who are doing substantial amount of work in that domain. Okay, so by studying the review, you will get a chance to study all these within a one manuscript. Okay, to get idea within by simply reading the one manuscript, you will get enough idea about that particular domain. So always I insist them, after reading the books, I always insist them to read the review articles, and finally they should read the original research papers, because original research papers are quite relevant to your study, to your hypothesis, to your plan of work. So original research work is in alignment, always should be in alignment with your study, okay, with your data, with your analysis. So these are the primary sources, I strongly believe, with the original research papers, the manuscript, the patents, or the analysis in and around the your research project is the primary sources. Secondary sources, as I refer, maybe a board overview or the overview what you have done in your research family, systematic reviews that you have read, again some books that you have read. So all these are the secondary sources. So always remember that both sources are very important, and you should have always a balance approach for reading the books, for reading the reviews, having some overviews on one hand, and other hand original research articles, the patents, and etc has to be read, and then you will have a clarity for that research project. Identifying the research gap is very important. Again, like for example, one hypothesis where I still remember I went for one Viva, PhD Viva. The hypothesis was like encapsulating a drug in a nanoparticle and checking its viability. Okay, so I think there is no gap. So gap has to be understand first. Maybe a research gap, maybe a market gap, maybe some knowledge gap. So how to identify and formulate the research question? It is totally based on, if you are doing some innovations for the existing product or existing technology, you should first understand what is the gap. As I mentioned earlier, I gave you the example of my PhD student who is working on the anti-epileptic nasal spray for the brain targeting. So there are so many products available, but those are oral pills. I mean, tablets are available. More often than not, physicians are prescribing the tablets which are having some issues like gastric disturbances, the first pass effect, the viability issues, and whatnot. So targeting or how much quantity is reaching to the brain, it's very very less and it's quite insignificant. So directly approaching the brain, targeting the brain via nasal spray is quite a good idea. So we have identified the research gap for that matter. So literature review to find the unexplored area is the first thing. And if not, if the area has already been explored, okay, इट इज नॉट ऑलवेज लाइक दैट कि हम बोलते हैं ना कि सर हम जो भी आइडिया हमारे दिमाग में आती है हम डालते हैं तो उसके ऊपर ऑलरेडी काम हुआ है, ऐसा होता है बहुत बार. बट सी it is not the only thing to think about. Other way we can think, for example, if there is product available, what is the gap with that product or existing range of the product. Existing technology है but उस technology में कुछ gap है क्या, that gap can be addressed. It is also a research. So addressing those gaps, identifying those gaps and formulating your research question around that gap, okay, is very good concept, I would say. So create a clear focus and researchable question, formulate research question which should be clear, which should be focused, and it can be around the gap. Okay, research gap can be identified through literature review, through understanding the market dynamics, consulting the physicians. So I always recommend my students to consult the stakeholders, okay, different stakeholders of the researcher. Now the question must be arise in your mind कि sir stakeholder यहाँ पे क्या कर रहे हैं, stakeholder research में क्या करेंगे. So there are different stakeholders in the research also. Who are those stakeholders? If I would like to work on a anti-epileptic nasal spray, okay, I shall first contact or have a communication with the clinician, the physician, because he is one or she is the one who is prescribing the medicine day in and day out, and they can tell me about which are the leading molecule I am referring. Okay, I am prescribing and what are the gaps over there. ये मैं molecule हमेशा लिखता हूँ, ये drug मैं हमेशा लिखता हूँ, prescribe करता हूँ, लेकिन इसमें मुझे ऐसे problems आ रहे हैं. Viability is very less, the dose patient compliance is very less, gastric irritations आ रहे हैं, other problems are there. So those gaps will only be understand by consulting the stakeholders. Different stakeholders are there for the different types of the research. Your mentor, right from your colleague, okay, who are working with you, your classmate, your fellow, class fellow, your group mate, your roommate. So all these can be considered as the stakeholders, because sooner or later you are going to announce some result in the public domain and they will be reading that. If not, if you are developing some innovative product, then also there are so many stakeholders available in the market. So you need to be very vigilant and you should be very approachable to those stakeholders to get that market dynamics. Even if you go to any pharmacy, even a wholesale pharmacist, if you approach, he can tell you about how many number of products it is available for that particular disease. Okay, so for that matter, you have to approach all this and mitigate the gap. It is not like that कि unexplored area ही हमेशा research के लिए हम ले पाएंगे. It is not the case, because it's hardly possible with the advent of the technology. If you if you have formulated any question, if you have generated any research question, and if you Google it out, you will get one or more paper. So you can you will be frustrated at times कि सर जो भी मैं कर रहा हूँ तो ऑलरेडी अवेलेबल है. तो ऐसा नहीं है, जो अवेलेबल है उसमें से गैप ढूंढो, उस गैप पे भी काम कर सकते हो आप. सो दैट कैन बी डन अगेन, व्हाट आई मीन टू से. So then finally hypothesis development that we have already discussed, and finally summary of my this discussion is about the integrity, identity and impact. I always insist on confronting the reproducibility with the transparency and in the open science. Whenever you are in science, your credibility will depend on whatever you are doing and how you are doing and how you are disseminating. Okay, it's very important to have an integrity. Integrity with the data is very important. If not, you will suddenly lose your credibility. If you are doing some manipulations around, if you are publicizing that data to the any other journals, predatory journals, what happen? Your credibility will not be developed, your goodwill will not be developed, and nobody will recognize you, identify you as a good researcher. So if you want to create your credibility, so these three things are very important. If you would like to build your identity, those two things are very important. Integrity with the data, ethics is very important. Do the thing ethically, don't follow the shortcuts. Okay, whatever you are doing, follow it judiciously, do it meticulously, and generate the data and follow that data only. If not, if you are not getting the good data, the results are negative sometime, then you can repeat that, try to reduce the error, but do not compromise on the ethics front. Do not compromise on the data integrity front, because if you are doing that, one day surely you will lose your credibility. Nobody will ask you again, and you will lose your recognition. So, and finally, impact. Whatever you are doing, whatever research you are doing, it should be impactful. Okay, it should be translational, it should be meaningful, it should be viable. So these things always you should keep in your mind. Research is not just an academic exercise. Okay, it is a backbone of I would say any advancement. Okay, so as I was giving examples, so many examples I gave to you. I told you that right from the vaccine development to the nanomedicines, every innovation starts with a well defined problem and a structured scientific approach. Then only a meaningful research can be done. I was reading one quote recently on some social media. Yeah, a take-home message is a PhD is not just a project to be completed and written up, but it is a training period during which aspiring researcher or scientist must learn the right way to do the science. So this is very, very good quote or the take-home message which I would like to convey to you people. And with this note, I rest my case to all you. If you have any questions, I would be more than happy to answer those questions. Thank you very much. It's over to you, sir.
Yeah. Sir, really, this is a wonderful session. What exactly the research? So the session is open for the
Yeah.
question.
If somebody would like
So,
to ask, they can also,
yes,
ask in the
really
chat
you have
box.
to ask. There are some questions in the chat box also,
Yeah.
but I requested the participant, please unmute your microphones and ask the question. No doubt.
सर जी
Yes.
सर
Yeah,
मुझे ये
continue,
पूछना है
sir.
हाय यस सर मुझे ये पूछना है हमारे जो रिजल्ट आए हैं सर वो निगेटिव में आए हैं तो निगेटिव रिजल्ट्स भी पब्लिश हो जाते हैं क्या सर
होते हैं ना, there are some journals or journals of the negative results, so you can publish that again. But before going to that, I recommend you to just have a thorough review with some of the stakeholders. As I mentioned, you can approach to some expert and who could actually help you to recognize, to identify what was exactly problem while doing that, while following that plan of work or doing that experiment. Maybe some error may be there, and because of that only you are getting those negative results. So before going publicizing those negative results, I recommend you to please have a review and have a discussion around your research family, so that you could come out with some solution, and then again positive results may come out of that. If not, there are some there are some journals available to publish the negative results also.
ओके थैंक यू सर
Yeah.
I requested Vishnu Jadhav, sir. Please ask the question. Sir, in the chat box, already the question.
Dr.
put up.
Vishnu sir has asked that please elaborate the application of nanopolymer in drug delivery, effective drug targeting. It's really nice question, Dr. Vishnu. As you were asking about the nanopolymer. See, there are so many polymer conjugates that can be used for the different drug delivery. So particularly, I would be able to answer you or guide you in this regard once your problem will be focused enough, because this is lacking the focus, nanopolymer in drug delivery or effective drug targeting, because there are so many areas wherein you can actually have some nano conjugates or the polymer conjugates. Recently I have conjugated one polymer and nanofibers we have developed, which was used for the MDR, multi-drug resistance bacteria. Klebsiella pneumoniae we tested for the ciprofloxacin. So we got a fantastic results, and that we tested on the biofilm catheter जो हम लगाते हैं इसमें हॉस्पिटल में, उसमें जो बायोफिल्म डेवलप होता है उसके ऊपर हमने काम शुरू किया, उस पे हमने बायोफिल्म का एक कोट लगाया, तो we found that the biofilm was not formed over there. So you can do this kind of a stuff with the nanopolymer conjugates in the drug delivery and drug targeting, of course. But I would be able to guide you in this regard once you have a clear-cut idea and a clear-cut question with a more focused question, I would say.
Sir, another question.
Yeah.
Please elaborate on the in vitro diagnosis kit and how to get the knowledge and hands-on experience.
Fine.
Now,
This
from
is
Son
a very, very good query for this matter. In vitro diagnostic kits are very, very costly, and always Thermo and Thermo Fishers with some Sigma Aldrich it is available. But if you have funding with you, go for that. If not, please collaborate. There are so many, in and around our Nagpur, there are so many labs who are actually eager to collaborate. For me and my research team, we always collaborate with the SIMS, Central India Institute of Medical Sciences, wherein there are so many science students. I found all students are all fellows are from the science background. No fellow is there from pharmacy background, sir. So in the SIMS itself, they are working. Your MSc PhD fellows are there as a project associates. You can get more knowledge from them. Which kit I should use, and what is you can get, of course, the pricing and all these. It's immaterial for that, क्योंकि आपको pricing वैसे भी मिल जाएंगे अगर आप Google out करेंगे तो. But आपके पास फंडिंग है क्या? अगर नहीं है तो फिर आपको अप्रोच करना पड़ेगा, कोलैबोरेट करना पड़ेगा, और उनके भी कुछ को इन्वेस्टिगेटर को साथ में लेके पेपर बनाना पड़ेगा या पेटेंट में उनका नाम डालना पड़ेगा, बिकॉज आपकी एक जो बाजू है वो थोड़ा सा कमजोर है, आपके पास फाइनेंस नहीं है, और सारे डायग्नोस्टिक किट आई थिंक it's cost around more than 50,000 any kit for that matter. सिंपल छोटी सी किट भी लेने जाएंगे आप तो 50,000 के अबव आएगी. तो आप you can get enough knowledge by searching these books product catalog from Thermo Fisher or Sigma Aldrich, for which you would like to have that diagnostic kit. And once you will be clear with the funding and the pricing of that kit, if it is not possible for you, you have to collaborate with some of the lab wherein those kits are available, and it's a routine practice for them. So that way you can do. And they also run some certificate courses to get the hands-on. So please be vigilant and be in a touch with them. कि जब भी ऐसा कुछ workshop हो, तो you call we people so that we can join and have a hands-on training. Otherwise,
Yes, sir.
I'll prompt Banginawar sir, Yogesh sir, whenever I'll have that kind of notification with me.
Yes, sir.
Yeah.
Thank you, sir. Thank
Yeah.
you so much, sir. I just wanted to ask you about this question only, in vitro diagnostic kits. Sir, you explained very well.
Thank
Very
you.
well. Thank you so much, sir. It
Mm.
will be the great help for me. Thank
Thank
you,
you.
sir.
Pleasure is all mine.
Yes,
Sir, another question.
Yeah.
Please suggest some laboratories where the drug delivery application, characterization, and performance in India for nanoparticle targeted drug delivery system.
Sir, our lab is fully equipped with the nano formulations, because I have all the facilities, like right from the probe sonication, then high pressure homogenizer, high shear homogenizer, again Zeta sizer, particle size analyzer, freeze dryer, everything is with me. So if you would like to work with me, I am open to work. So there are so many labs across the India, but again आपके उनके रिलेशन और आपका उनका मतलब डिस्टेंस कितना है. आप जैसे सर ने मुझे अलाइन किया नागपुर में माइक्रोबायोलॉजी के लिए, अदरवाइज वो खुद भी कर सकते थे वो काम, लेकिन सर ने मुझे बोला कि सर हम दूर है थोड़ा सा, तो आप नागपुर में काम कीजिए, इन लोगों के साथ में काम कीजिए. तो इसी तरीके से आपकी ये फिजिबिलिटी भी देखिए आप. वैसे नैनो का काम मेरे दादा साहब बालपांडे कॉलेज में सारा नैनो का काम हो सकता है, सारे नैनो के इक्विपमेंट्स है. नैनो ड्रग डिलीवरी पे हम काम करते हैं. हमारे यहाँ पे एक्सपर्ट्स भी है, 4-5 लोग हैं प्रोफेसर लोग जो नैनो पे ही काम करते हैं. वी डू हैव अ गुड पब्लिकेशंस आल्सो. इफ यू सर्च दैट स्कोपस डेटा और वेब ऑफ साइंस डेटा, यू विल फाइंड सो मेनी पब्लिकेशंस publications from our lab. Almost around हमारा जो impact factor है सर, अभी cumulative impact factor हमारा beyond 300 गया है. तो इतना काम हमने substantial काम उसमें किया है. 42 patents we have granted from last 3 years. So you'll find such labs. And of course, I take this opportunity to welcome you all if you would like to do any nano formulation work or any drug delivery nano therapeutics work. So you can collaborate with us.
Yes, definitely, sir. This is wonderful. Sir, my one question at the last.
Yes, sir.
So
Hello.
I read from the articles many a times. Whatever may be the plastic, that will be the waste one. Can it is converted into the any type of the pharmaceutical API?
Plastic.
Yeah, plastic.
Look,
I read more than 10 articles.
yes, sir.
They are
See,
converting into the medicine with the help of the certain catalyst. It is
plastic,
feasible
plastic,
one.
yeah, plastic converting to the medicines.
Yeah.
Sir, I don't have any idea about that. I need to read some articles around that.
Yeah.
So
But I
please-
read more than 10 articles which indicates one. The waste
so please
particle we
share
are
those
converted
articles
into
with
the
me. Please share those
medicine.
articles with me. I'd be more than happy to read those and get better understanding out of that.
Yeah. Sir, one
So my-
question.
Yes.
So one question.
So we are doing
Ashok
a
sir-
lot of work.
Ashok sir was asking something. Yeah.
We are doing a lot of work. When you are doing really a research in laboratories, can every time we get the feasible result?
See, for that matter, this question is quite aligned to the what earlier question was asked. What I mean to say, if you have planned it properly and if you have a good resources with you, resources I mean not only the equipments, not only the expertise, but also the publications that you are reading and that you are following. Okay, always follow Q1, Q2, Q3 journal, rank journal manuscript, which are available or in the public domain. And if you are following that rigorously with the quality equipment, with the quality chemicals and error-free execution, more often than not, I guarantee you will get a good results, visible results. Even
But,
though if it
many-
is not possible in that case, even if by following these things rigorously, if you are not getting the result, means that data is not integrated enough, or there are some manipulations around that. It does happen sometime, but rare case, I believe. So you can switch to other journal, to other publication, and you have to have so average of some 10, 14 readings. Okay, you cannot simply rely on any one manuscript. So that is my suggestion, groups.
Yeah. But many a times, can we run every experiment in a triplets manner?
Yeah, of course, of course. See,
But in a
whatever-
present
whatever
scenario, the
quantitative
students are
data you are
not
generating-
doing in such a manner.
Correct, sir. Correct. Absolutely. I do understand that whatever quantitative data you are going to generate, okay, if possible, you have to do it in a triplicate, n is equal to 3 at least, and then you have to have some standard deviation, standard error around that. Okay, so that
So I think that will
whatever
minimize the
quantitative
error.
data you should have some ANOVA, one-way ANOVA, two-way ANOVA, statistical application or design has to be there to validate those results. Because I said earlier, validation is very, very important.
Yeah, yeah.
Yes, of course. Yeah, Ashok sir, you were
So,
asking
any
something.
questions from the participants?
Ashok sir was asking
Sir,
something.
my question.
Yeah.
Yeah, yeah, please.
Go ahead, sir.
First of all, sir, thank you very much for your wonderful talk. My question is, sir, slightly different from your topic, but it is related to the research methodology. Sir, we all know that due to the introduction of AI, there is a transformation of research in the field like healthcare, microbiology, or pharmacy. Okay, many researchers are nowadays they are using AI tools for their literature review, methodology, or dataset analysis.
Mm-hmm.
So can this lead to the over-reliance,
See,
reducing the critical
see,
thinking?
yeah, understood. See, there should be a balanced approach. So AI is always there to help you out, and there are so many, so many time you can use the AI, of course, and it would be of your help. Meaning, whatever you are using, whatever software you're using, whichever AI you are using, it will going to help you out eventually. But you should not rely, as far as the data is concerned. Okay, see, there are two prominent applications of the AI. Like for one is a data-driven AI, and other is a machine learning AI, for example. So whatever data-driven AI is there, you cannot rely simply on that data-driven AI. But machine learning AI, it will definitely going to help you out, because those are the softwares, or those are the prompts that has been generated out of that machine learning, and then that will be helpful to you to proceed with your research, to proceed with your experimentation. So that is why I always recommend it should be a balanced approach. You should always cross check when it comes to the data, and if you have softwares, if you have some AI tools with you, there is no harm in using the AI. Okay, so instead of that, I recommend all of you to use more and more AI whenever it is due to be used or required to be used. Okay, if not hampered your data or data-driven AI, as I mentioned, don't rely simply, but machine learning AI can be used every now and then. There is no harm, and you can have a good, validation with or generation of the data by using the AI. So, Yogesh sir, one of my friend is there. You might have known about him, that Satish Pulshettiwar is one of the. Yeah, so he can be a good guiding point, or can be a good resource for how AI could be useful in basic science research. So you can arrange his talk, as today you are arranging this one. So
Yes,
I'll
definitely,
tell
sir.
him.
Definitely, I will
Yeah,
in touch with the Satish sir.
yeah.
Thank you, sir.
Sir, but when you are saying the AI tools, you have to apply it.
Yeah,
But many a times we can't use our brain.
of course.
Directly, we are jumping on the AI and start searching. It will be the ethically when you publish the certain papers. Most
No,
of the writing it is coming from the AI right now.
see, those journals which I was talking about, the Q1, Q2, Q3 journal, they are asking for the AI plagiarism also. There are two types of plagiarism nowadays. So you have
Yeah.
to check the
Similarity and
database plagiarism
AI
and AI
report.
plagiarism. So whatever you are writing with the AI help, that will not going to serve your purpose. Anyway, it will be rejected. AI generated जितना भी है वो plagiarism में आ जाएगा. तो उसमें कोई इशू नहीं है सर. जो भी यूज करेगा उसका पेपर रिजेक्ट हो जाएगा 100%.
So, any questions from the student? I think
Hello.
Rakesh sir is.
Hello.
Besen sir.
Sir, actually, I want to know. I have already made my materials and I have done so many characterizations. So, from this characterization, how I will know that it will be applicable for delivery systems?
Okay, see, based on the earlier literature, if you're after characterization, you have a data, and that data you have analyzed. If it is matching to the any other material which previously has been used for any drug delivery system, so you can always have an opportunity to use your material, because it has having a same characterization to that of a material which is already existing. So just compare case by case. Okay, so comparative analysis has to be done. Once you have a comparative analysis, and you'll be coming to know that this is a material which previously has been used with the same characterization, I have developed one material that can be used for the that particular drug delivery. So it's very simple, sir. Compare it with the existing materials which is available in the public domain.
Okay.
Yeah.
Thank you, sir.
Yeah.
Good afternoon, sir.
Good afternoon.
I am Dr. Rakesh.
Yes,
One
Rakesh sir. Sir, you are not audible to me. Sir, is he audible?
Am I audible, sir?
Yeah. Now you are
Yes,
audible.
yes.
Yes, yes.
Sir, I am Dr. Rakesh Thackeray from Yashwantrao Chauhan College, Lakhandur, and one of the organizer of the event. And I would say, sir, we are very fortunate to have a speaker like you.
Thank you, sir.
Don't take it
Pleasure is all
otherwise.
mine. I am fortunate rather.
Don't take me otherwise, and definitely you will agree with me. Most of the pharmacy persons are, you know, they have big ego. But I have seen today the person with the pharmaceutical science having good research attitude, scientific temperament, and less ego or without ego. So
Thank
I would like to mention this quality from your side. And that's why I said we are very fortunate to have the Dr. Nilesh Mahajan
Rakesh
with
sir,
us.
I take this opportunity to welcome you at my institute. I
Sure,
show,
sure.
yeah.
I'm
I
going to mention
show,
this.
I show you all my facilities to you, and
Right, sir, right,
you are
sir.
always welcome to use those facility. Don't bother about that, and I can also meet you with my colleagues who are there to help you out anyway, anytime.
Sure, sir,
Okay.
sure. I was there throughout your talk, and I listen each and everything, and that's
Thank you,
why I
sir.
mentioned this thing. You are pharmaceutical scientist without ego. You mentioned earlier, anyone of from the participant can come to your place, and you will collaborate with them.
That is the reason, sir. That's I have a good collaboration with the basic science as I am doing some stuff with the Porwal College with
Right, Alok Nayak.
Rai sir.
Alok Nayak. He is
Alok Rai
our
sir.
senior.
With
Yeah.
Ramdeva College of Engineering, I have 4 patents. With Raisuni College, I have patents. Then Dental College, I am doing NVC, Nagpur Veterinary College. I am a research advisory committee member. So
Right.
I have good collaborations with Department of Physics, with Department of Chemistry. I have published a paper. LIT also.
Yes.
There is a one book I can show you. This is a book, sir. This is an international book. Okay. And this is a nanophysics and photophysics. Can you read it?
Yes, sir. Sure.
Yeah. So this
Okay.
the authors अगर आपने देखेंगे सर इसमें तो authors इसके जो है ना वो डिपार्टमेंट ऑफ फिजिक्स वाले ही है आपके
हाँ, सर.
डोबळे सर ठीक है ना
Right,
तो
right.
वो भी मेरे कोलैबोरेटर हैं सो व्हाट आई मीन टू
Right,
से
sir.
कि आपने जैसे आपने बोला कि इफ यू हैव अ if you don't have ego okay you will find so many friends.
Yes, sir. That I would like to mention your quality.
Thank you, sir.
And definitely in future we will collaborate for the same. Actually, I have also ready to submit my minor project, and I have one more month. I will communicate with you, and we will try to register a patent for the same.
Of course.
Sir, besides this, I have
3 days, sorry to interrupt you. 3 days back itself, I got one patent with Nagpur Veterinary College, sir, which was pending from 2018 itself.
okay.
Yeah.
Actually, I was working on the metagenomics and metaproteomics.
Good,
So
sir.
I will communicate with you, and we will try to plan for a
Fantastic,
patent.
sir. Lots of
Thank
learning
you.
will be there from you. So I'm
Sure.
eager to
I'm not such.
I'm eager to talk to you more on this topic more and more.
Sure, sir. Sure. I will be there at your place very
Pleasure.
soon.
You are always
I have
welcome, sir.
one more questions regarding this invited talk or research methodology question. As we have participants from arts, commerce, and science faculty from our college, also our students are there. So I would like to ask a one question. As scientific person have to publish their research through Web of Science, Scopus, elsewhere, Springer Nature. What type of the special journals are available for these humanity background people? Are there any Web of Science
Humanities
journals?
is there, sir. If you refer the Web of Science data, just Google
Right.
it out, Web of Science data, and into the subject area, just drop down box, you click the humanities, you'll get all the journals. Yeah.
They are also having the Web of Science journals.
Of course. That's why it is mentioned in the Web of Science data.
Right. So,
Yeah.
Thank you, sir. Thank you
so
very much.
आणखी सांगा सर
Thank you,
योगेश सर.
sir.
Shall I leave?
So at the end, without these formalities, the session should not be an end. That will be the vote of thanks. First of all, thanks to our principal of both the college, Dr. Hadge sir, Dr. Dadve sir, who inspire every time to do something for the student, a development as well as the faculty members. I really thankful both the principal. I thankful to Dr. Wasnik sir, IQAC coordinator Yashwantrao Chauhan, college Lakhandur. I thanks to Dr. Davande sir, IQAC coordinator Shankarrao Bhoyer Patil Mahavidyalay Pulgaon. I thankful to Dr. Rakesh Thakre sir, and very important, but at the last one, I thankful to Dr. Nilesh Mahajan sir, today's resource person, who explained in a very wonderful way what the basic research are there, what are the different steps are there, how the topic should get a selected one, and really what will be the inputs and what will be the outputs, and it should be a ethical one. I really thankful to Dr. Nilesh Mahajan sir, professor and head Department of Pharmaceutics, Dada Saheb Balpande, College of Pharmacy, Nagpur, who spare his holiday with us.
It's not holiday for me. I am at college only.
Okay. So I really thankful to sir for
Thank you,
your
thank you.
wonderful explanation on the research methodology.
Thank you,
I
sir.
also
Thank you so
thankful
much.
to all the participants, faculty members, IQAC coordinators, associate professor who are joining today's session. On the behalf of, under the aegis of IQAC and Department of Microbiology, I really thankful to everyone. I also thankful those, particularly the person who are helping behind the curtains for at the permission of today's resource person. I close the meeting. And definitely we are meet in the offline way, sir.
Looking forward.
Yeah.
Looking forward to meet you all offline, in offline zone.
Yeah, yeah,
Sure, sir.
definitely, sir.
Sure, sure.
Definitely, we will be having collaboration with you.
Of course, of course.
We are also eager to have collaboration with.
Yeah, love to have collaborations.
Really nice experience to attend this program, sir.